This chapter will be on sex determination and sex-linked characteristics. First, we'll talk about how sex is determined. So most of you are probably familiar with the mechanism of sex determination in humans, and this is an example of a chromosomal type of sex determination. So any individual who gets two X chromosomes is going to be female, and an individual with an X and a Y chromosome will be male. This same system is used in most mammals and also in a lot of insects. And so Drosophila, the fruit flies that we are using in lab, also have this XX and XY sex determination system. Some other chromosomal sex determination systems exist, however. It's not all the XX versus XY. For example, in birds, if you have two Z chromosomes, you're going to be a male. If you have a Z and a W chromosome, you'll be female. There are also other types of sex determination based on the inheritance of a particular gene or based on some environmental situation, such as temperature, which is found in turtles. We're going to focus primarily on chromosomal sex determination, and I'd like to just talk about these terms at the bottom. First of all, if we're talking about a sex chromosome, that would be the X and the Y, or in the case of birds, the Z and the W. In comparison, autosomes are all other chromosomes. So any chromosome that is not a sex chromosome is going to be called an autosome. We'll also talk about the homogametic sex versus the heterogametic sex. And what this means is just how many different types of gametes you can form. So for the homogametic sex, in humans, that would be female because they only make one type of gametes. All of them are going to have an X chromosome. In contrast, males are the heterogametic sex because they make gametes with an X chromosome and gametes with a Y chromosome. We'll also use the term hemizygous, and we'll use this when an individual has only one of a particular chromosome. So normally, if we talk about the alleles of a gene, we would use the terms homozygous or heterozygous, and those two terms, neither of them really apply if you only have one of that particular gene. So this is an exception to Mendel's rule of genes come in pairs. Males only have one of each gene on the X chromosome, and we would say that they are hemizygous. Now, in the heterogametic sex, in human males, for example, the question arises of how those sex chromosomes segregate properly during meiosis. So if you recall in meiosis 1, the homologous chromosomes line up together and form a tetrad, and then they segregate, one to each daughter cell. So it turns out that if you have an X and a Y chromosome, that the X and the Y will actually pair together as if they are homologous to each other during meiosis 1. The reason that these chromosomes can pair with each other is because they actually share these regions of similarity that are called pseudo-autosomal regions. So these are found at the ends of the Y chromosome and the X chromosome, and they allow the X and the Y to line up and then segregate. So the X goes one way and the Y goes the other way. So next we're going to talk about sex-linked traits that are determined by genes carried on the X and the Y chromosomes. And I'd just like to say a little bit about the two sex chromosomes in humans. So the X chromosome is a really big chromosome. It has a lot of important genes on it, and at least one copy of an X chromosome is required for life in humans. In contrast, the Y chromosome is not required for life, obviously, since you have females without one. And interestingly, there is one particular gene that is on the Y chromosome which determines maleness, and that gene is called the SRY gene. In addition, there are other genes located on the Y chromosome, and these genes are primarily important for spermatogenesis. So it tends to be that if you have a mutation on the Y chromosome, you tend to be sterile because you cannot complete spermatogenesis. So we're really going to focus mainly in humans on X-linked traits because these are the ones that are much more common. One thing I'd just like to mention is something called dosage compensation in mammals. We will come back to this later in the course, but this is discovered in 1949 by Murray Barr. And what he noticed is that in female mammalian cells, there is something in the nucleus, and he called it a Barr body. It turns out that this is one of the X chromosomes that has been inactivated. And the reason why it becomes inactivated is to equalize the dosage of each gene on the X chromosome between males and females. So we have roughly the same amount of protein being produced by each gene on the X chromosome between males and females. And since that X inactivation occurs very early in development, you can have a heterozygous female that actually looks mosaic, that some cells have one X chromosome being active, other cells have a different X chromosome being active. This is the reason why we actually observe the coat color of tortoiseshell cats. So X-linked inheritance was first discovered in Drosophila by the lab of Thomas Hunt Morgan. And this was very soon after Mendel's work was rediscovered, so early, about 1910, 1915. Morgan's lab was actually using Drosophila, and they were isolating different mutants, variants from the normal wild type. And one of the mutants they isolated was a fly that had white eyes instead of red eyes. So they expected a certain inheritance pattern based on Mendel's predictions, and they found that there were some really unexpected results. If the white-eyed mutant was actually a standard autosomal inheritance pattern as described by Mendel, what we would expect is, since the white eye allele, which we're going to call white minus, is recessive to the white plus, which is the dominant wild type allele for red eyes, we'd expect that if you cross a white-eyed fly together, homozygous true breeding line, together with a homozygous red-eyed fly, you would get all red-eyed F1 progeny. What they actually found instead was that the progeny depended on the sex of the parents. So if they crossed the females that were red-eyed and the males that were white-eyed, they found that all the progeny had red eyes. So just as expected with an autosomal inheritance pattern. However, if they did the reciprocal cross, so that means the male now was the red-eyed parent and the female was the white-eyed parent, what they found is that in the F1 generation, all of the females had red eyes and all of the males had white eyes. So this trait of white eyes is somehow linked to the sex of the organism. This discovery eventually led to the understanding of sex-linked traits and how the X and the Y chromosome function to determine the sex of the organism. So I'd like to end up by showing you how we will write out crosses with sex-linked traits and show you how we'll do the Punnett squares. So we will include some additional information. We will always indicate what kind of chromosomes are present, and we will indicate which kind of allele is present by showing it as a superscript. So for this cross we just talked about that Morgan did, the red-eyed male is going to be an X and a Y. And since they've got red eyes, we're going to have a white plus allele on that X chromosome. For the white-eyed female, the female will have two X chromosomes, and both of those W genes will be white minus alleles. When we do this cross, we will simply make a normal Punnett square. And first we're going to consider what types of gametes will come from each of the parents. So the gametes from the male are going to be either an X chromosome with a W plus or a Y chromosome. The gametes from the female there's actually only one kind, which would be an X chromosome with a W minus allele. So if we then fill this in on a Punnett square, the gametes from the female parent, there's only one kind, so we can just make one column here. And the gametes from the male parent would be either the X chromosome or the Y chromosome. We're going to join these together, and what we find is all of the female progeny, which have an XX, are going to be red-eyed, just as we predicted, and all of the male progeny are going to be white-eyed because they got that W minus allele from their mother.